So today's webinar is titled Zeptomole Detection of Protein Biomarkers in Serum Using an SPRi Nano-Aptasensor. We generally highlight the flexibility when we talk about our SPRi instruments, and certainly that's going to play a part in our speaker's ongoing research. But today we're going to be focusing on her approach to maximize its sensitivity, and that's what the Zeptomole is referring to. So looking at our list, there's some names on here that I recognize, and certainly some new names as well. So if you're new to Horiba or new to our instruments, I will briefly tell you what sets us apart from standard SPR experiments. As you'll see in Marinella's presentation today, we're imaging a flat gold surface with a CCD. And this allows us not only to multiplex and follow hundreds of binding events simultaneously, it also provides an open canvas for creativity. So for example, we can combine our chips with fluorescence, electrochemistry, insert them directly into MALDI mass spec, and adhere to custom microfluidics. During our broadcast, if you experience any technical problems viewing or hearing the presentation, please use either the chat box or the question box, and those should come directly to me. I'll try to work with you and get things cleared up. At the very end of the presentation, we're going to have a question and answer session. It's a live question and answer session. So our first broadcast this morning, we had many more questions than we could handle during that time. And if we don't get to your question, I apologize. We will follow up with you afterwards to either start a dialogue or get you an answer. All right, with that, let me introduce our speaker for the day. This is Dr. Marinella Sandros. She's an assistant professor at the University of North Carolina at Greensboro. She received her doctorate from Wayne State University under the guidance of David Benson, and she studied bioinorganic chemistry there. From there, she was a postdoctoral fellow at McGill University's Biomedical Engineering Department, working with Dr. Miriam Tabrizian. And over the last 12 years, Marinella has been developing new nanomaterials for fluorescence, Raman, and SPR, and continues to find new applications for them that extend the capabilities of techniques. It's too bad that you're not seeing her talk in person because she's very animated, so you can maybe imagine lots of hand gestures as you hear her speak. With that, I'll unmute our speaker and let her get started with the presentation. Marinella?
Thank you, Jeremy, for a wonderful introduction. I'm very delighted to tell you about the SPRi Nano-Aptasensor platform we have developed in my laboratory. And also, I would like to take a moment and welcome all our attendees that are with us today. Briefly, I will cover the following topics. I will start off with a general explanation of SPRi technology and relate how SPRi can be a useful tool for biomarker detection. In addition, I will spend a little time comparing ELISA to SPRi. Finally, I will discuss the construct of our proposed ultra-sensitive SPRi platform. Before discussing surface plasma resonance, I need to introduce the concept of total internal reflection. So when you have a light beam hitting a prism, the light is bent towards the plane of the interface. When it's passing from a denser medium, being here the glass, to a less dense one, which is present here as the air, this is known as refraction. Changing the incident angle changes the outcoming light until the critical angle is reached. At this point, all the incoming light is reflected within the prism. This is called total internal reflection. So the light cannot exit the medium and is effectively trapped in the denser medium. Surface plasma resonance is a powerful technique to measure biomolecular interactions in real time in a label-free environment. SPR is an optical technique that measures the refractive index within 300 nanometer of a sensor surface. Now, surface plasma is basically electromagnetic wave propagating along the surface of a thin metal layer. And here in our example, we have gold. Excitation of surface plasma is based on total internal reflection. This means when an incident beam of p-polarized light strikes an electrically conducting gold layer at the interface of a glass sensor with a high refractive index, an external medium having here we have is water, which has a lower refractive index, and at a given angle the excitation of the surface plasma takes place, resulting in a reduced intensity of the reflected light due to resonance energy transfer between evanescent wave and surface plasma. Basically what happens is the photons are converted to plasma, or we can say free electrons at the surface of the biochip absorb incident light photons and converts them into surface plasma wave. As shown to your right, a dip in the reflectivity of the light is seen under these SPR conditions. The angle at which the resonance occurs in our example, theta 2, is extremely sensitive to any change in the refractive index of the medium adjacent to the metal surface, and such changes can be monitored by recording the intensity of reflected light when the system goes out of resonance. This is the basis of surface plasma resonance measurements. The resonance conditions are influenced by the material adsorbed onto the thin metal film. There's a satisfactory linear relationship between the resonance energy and the mass concentration of the biochemically relevant molecules, such as proteins, sugars, and DNA. The SPR signal, which is expressed in resonance units, is therefore a measure of mass concentration at the sensor chip surface. This means that the analyte and ligand association can be observed, and ultimately rate constants as well as equilibrium constants can be calculated. While, as shown here, one of the interactants is immobilized to the sensor surface, the others are free in solution and pass over the surface. Association and dissociation is measured in arbitrary units and displayed in a graph called the sensogram. Biomolecular interaction analysis is not limited to proteins. Interactions between DNA-DNA, DNA-protein, lipid-protein, and hybrid systems of biomolecules and non-biological surfaces can also be investigated. So a lot of times when I mention that I work with SPRi, I always get the question, well, we've heard about SPR, but what is the I for? And I say it stands for imaging. However, truly, to me, it means imagination. Why I say that? Because I view SPRi biochip like a white canvas that I can paint in any way I please. This, in turn, gives me a lot of freedom and flexibility in my experiment design. The SPRi imaging technology takes SPR analysis a step further. It allows you to visualize the whole biochip via a video CCD camera. The high-resolution CCD camera provides real-time difference images across the array format. Now, when you're setting your experiment, you have the liberty, as you can see here are your spots, you have the liberty to define each spot or region of interest up to 400 spots and monitor activity of each spot at real time. As shown after a target in our diagram here, after a target analyte binds, you observe a change in reflectivity in the sensogram to your right. And all the spots where binding occurs light up, which is captured by the digital image. Now, as opposed to systems that use independent channels for parallel measurements, the SPRi allows you to define positive and negative controls whose measurements are collected at the same time and under the same condition as the experimental samples. I would point out that SPRi platform is not limited to biomedical research, but also has found applications in food science, environmental, and agricultural science. In the next slide, I will walk you through the SPRi system. Here you'll find this is the SPRi Plex 2, and here you'll find the smooth flow pump, and typically your flow rate would be around, I would run around 50 microliters per minute. And over here is your injection valve. Typically we inject 200 microliter. You can go higher to 500 microliter. This is depending how much sample you have, and also you can go lower depending on which instruments you have. The temperature control allows you to run your experiments at temperatures between 15 to 41 degrees Celsius. And here's basically a schematic of how the optical system looks inside the instrument. And right here is where you insert your biochip, basically a prism that's coated with a very thin metal, very thin layer of gold. And here's your flow cell. This is an illustration of the SPRi optical system. So an LED source is sent towards the functionalized gold surface through a high refractive index prism, resulting in the excitation of the surface plasma. And after reflection on the chip, the beam is intercepted by CCD camera, where it collects the output signal. The definition of a biomarker is an indicator of a biological state. In a sense, it relays information about a change in expression or state of a protein that correlates with the risk or progression of a disease. Blood-based biomarkers are not limited to proteins, but also can be DNA or RNAs, microRNAs, peptides, and cells. Since the molecular changes that occur during tumor development can take place over a number of years, biomarkers potentially can be used to detect cancers early, determine prognosis, and monitor disease progression and therapeutic response. Detecting any disease early, including cancer, is very important because it usually means more opportunities for different interventions and therapies, with increased potential for improving patient survival and quality of life. For a biomarker to be clinically useful, analytical methods must be available that allow reliable measurement, optimally with capability for high throughput tests, prompt turnaround time, and reasonable cost. Current immunoassays measure proteins at concentrations above 10 to the negative 12 molar, whereas the concentration of the majority of proteins important in cancer, neurological disorders, and the early stages of infection are thought to circulate in a range from 10 to the negative 16 to 10 to the negative 12 molar among the sea of other biomolecules. So basically when I talk about this, it's basically like trying to find a needle in a haystack. There's a challenge because your biomolecule, the biomolecules that you're trying to detect is amongst other biomolecules that look very similar to it. An example of commonly used immunoassay is enzyme-linked immunosorbent assay, better known as ELISA, which is a test that uses antibody and color change to identify a protein of interest. ELISAs are performed in a 96-well plate, as shown here, which permits high throughput results. The immunocapture antibody are coated and dried in the microplates, and samples are then incubated in the wells. The target protein is thus bound in the well by the capture antibody, after which the unbound sample material is washed away. A second detector antibody is then added, which binds to a different epitope of the immobilized target protein. This detector antibody is labeled with biotin, which provides an attachment point for some kind of label. In this particular assay, they utilize horseradish peroxidase as a label, which is tagged with streptavidin. A substrate like TMB is then added, where it's converted by HRP into a colorful product, which can be read by most standard spectrophotometric plate readers. Now, high throughput and sandwich-based analysis are complementary features between ELISA and SPRi. High throughput means screening a large amount of samples, and a sandwich assay is when your target is sandwiched between a capture biomolecule and a detection biomolecule. I would like to point out that ELISA is a very good complementary technique with SPRi. However, SPRi can provide you with additional features such as real-time monitoring, multiplexing, which means the ability to detect multiple analytes simultaneously, and label-free analysis, where you don't have to label your analyte. Now, to me, the most attractive feature about SPRi for me is the multiplexing. This is very, very attractive, as the current literature stresses the need for simultaneous biomarker panel monitoring to improve accuracy of diagnosis. So basically, the ability to detect panel of biomarkers can better help clinicians identify or can identify disease. So there are two established techniques for biomarkers detection with SPRi. One is a label-free method where you directly detect the analyte, as shown to your left here. However, if your analyte concentration falls below the limit of detection of the instrument, you need to resort to a sandwich-type assay in order to amplify the SPRi signal, as shown to your right. Here's an example where they use gold nanoparticles tagged to detection antibodies, and the amplification occurred as a result of the coupling between the localized surface plasma resonance of the gold nanoparticle and the propagating surface plasma resonance of the gold film. Observing that there was a lot of work focusing on using gold nanoparticles for SPRi signal enhancement, I became very curious what would happen if we introduced quantum dots. The reason I chose quantum dots is because I've been working with them for a long time, and early on in my research career, I realized they are very good communicators. Now let me take a moment to explain to you what are quantum dots. Quantum dots are a special class of semiconductors which range from 2 to 10 nanometers in diameter. Because of their unusual high surface-to-volume ratio, quantum dots display unique optical and electrical properties that are different in character to those of the corresponding bulk material. The most apparent of these is the emission of photon under excitation, which are visible to the human eye as light. Moreover, the wavelength of these photon emissions depends not on the material from which the quantum dot is made, but its size. So here, if you're looking at the image, you see that the smaller, you can take a quantum dot composed, for example, cadmium, which are most commonly used, and you can take an 8 nanometer quantum dot and then you shrink it in size, and you can see the emission shifting to the blue. And this is, the material is not changed, it's just simply a size-dependent emission. And as you can see, they're extremely bright fluorophores too. Now, a good way to understand the special optical and electronic properties is to look at their bulk counterparts. So in this example, we're comparing cadmium bulk semiconductors to cadmium quantum dots. The bulk semiconductors, their band gap is fixed. It's a fixed parameter, and it's only determined by the material's identity. However, the situation changes in the case of quantum dots because of their small size. The electrons in the quantum dots are confined in a small space. And when the radii of the semiconductor nanocrystal is smaller than the exciton-Bohr radius, and the exciton-Bohr radius is the average distance between the electron in the conduction band here to the hole that is left behind in the valence band. When there's that distance changes, there's a quantization of the energy level. And what that means, the energy levels becomes discrete, and they become now related more to an atom. And this is why they have been nicknamed as the artificial atoms. I'd like to point out that the bulk semiconductors, their energy levels are continuous, whereas the quantum dots, the energy levels become very discrete and more separated from one another, and this is why it resembles an atom. Now, generally, as the size of the crystal decreases, the difference in energy between the highest valence band and the lowest conduction band increases. More energy is then needed to excite the quantum dot, and concurrently, more energy is released when the crystal returns into its ground state, resulting in a color shift from red to blue in the emitted light. As a result of this phenomena, quantum dots can emit any color of light from the same material and simply by changing the quantum dot size. So now I will introduce my early work with the SPRi platform. Here I collaborated with Drs. Malich and Tabrizian, and we investigated if quantum dots can enhance the SPRi signal. To your left is the design of our platform. We deposited sialylated single-stranded DNA onto the gold surface, and followed by the addition of heptadecafluoro-1-decane-thiol, which acts as a blocking molecule. And then we introduced the biotinylated complementary strand, and the concentration was 50 nanomolar. We observed a 2.5% change in reflectivity. However, after the introduction of the streptoavidin quantum dots, we saw a 10-fold signal enhancement. And this is where we injected, the point where we injected the single-stranded DNA. We saw a 2.5% change. And then when we added the streptoavidin quantum dots, you can see how the signal was amplified. This figure shows the SPRi binding kinetics for various concentrations of biotinylated DNA target sequence. The target was injected into the flow cell over here for 8 minutes, followed by a buffer wash and a 10-minute incubation with the streptoavidin quantum dots. The corresponding concentration gradient is plotted where each point represents the average value of the reflectivity difference between initial and final buffer injections, calculated from 3 SPRi kinetic curves. For comparison, the concentration gradient curve is also plotted for DNA hybridization assay without the amplification step in red. The limit of detection without the amplification step lies between 1 to 10 nanomolar for single-stranded DNA complementary sequence concentration. Conversely, using the quantum dot amplification strategy, an improvement of more than 6 orders of magnitude in limit of detection and 25-fold signal enhancement is obtained. The minimal DNA concentration corresponded to 100 femtomolar after an 8-minute hybridization reaction time. In addition, the use of quantum dot provided the means without any additional surface treatment to combine SPRi technology with fluorescence imaging. At the bottom, you see 3D images of the DNA sandwich assay biochip surface were reconstructed from Z-sectional images and were loaded into an Imaris 6.3 imaging software. So then we wanted to assess if quantum dots enhances SPRi detection and this can occur with a different biomarker. And the biomarker we chose is prostate-specific antigen. So the biosensor was functionalized with a mixture of sialylated PEG that has carboxylic group on one end, and another mixture of the PEG with OH group at the opposite end. Looking at the sensogram, you can see we injected PSACT in spiked serum and allowed to bind to the sialylated PEG that has the capture antibody bound to it. And then we added the PSA, then we followed by an amplification with the detection antibody. And then we added the streptoavidin quantum dots. And as you can see here, that direct antigen detection is incapable of distinguishing between the antigen binding and control. And we use an antibody that binds to IgG as our control, while the sandwich assay allows a specific detection of the PSA-CT complex at the concentration of interest. Now the corresponding difference image shows time-lapse binding kinetics for the initial buffer, which is around here, so at T0. And here's the injection of the PSA-CT, and you can see that binding is to your left. Let me point out is our target antibody that binds to PSA-CT, and this is our control antibody. And then you can see that both sides light up, as also seen as a large change in reflectivity here. And then after the buffer wash, you can see the spots are dimming. And then you can also see clearly that only to the detection antibody is only binding to your PSA-CT antigen, or to your experimental side, and not binding to the control. The addition of the quantum dots, you can see them highly illuminated. This correlates directly to the change in reflectivity. And after the buffer wash, you can see that it's still illuminated. So we've showed that QD's SPRi signal enhancement is a universal, meaning it can enhance the detection of various types of biomarkers. However, we wanted to probe how is the signal enhancement is occurring, and so we decided to assess quantum dots that emit at different wavelengths. Our first hypothesis is it just a mass loading effect, or could there be another phenomena that is occurring? If you look at the 800 nanometer quantum dot compared to the 780 nanometer, which are there's very little difference in size, however, we see a marginal difference in their amplification signal. This in turn revealed that there could be something else going on. And after looking through the literature, there were reports that the interaction of quantum dots and propagating surface plasma generated from a silver nanowire were bidirectional. So what they explained is that propagating surface plasma were able to excite excitons, which resulted with the quantum dot emission, and by direct excitation of quantum dots, the exciton decay got converted to propagating surface plasma. This translates to a coupling phenomena that is occurring between quantum dots and the propagating surface plasma. We believe the signal amplification is attributed partially to a mass loading effect, but in addition to the surface plasma generated from the gold surface exciting the quantum dots, which then the quantum dots transfer their excitation energy by radiationless transition to the gold resonance nanosystem. Few important conclusions we were able to make from our early work is that the surface chemistry influences SPRi detection sensitivity and amplification, and that the 800 nanometer quantum dots experience more signal amplification as a result of more coupling occurring with the propagating surface plasma resonance. So my current research focuses on developing a platform that can detect biomarkers present in serum at ultra-low levels, which I mentioned earlier is between 10 to the negative 16 to 10 to the negative 12 molar. A few things I learned also from my previous work is that the closer your amplifier or detection molecule to the sensor surface, the greater was the enhancement, as we saw with DNA in comparison with antibodies. I decided, as opposed to antibodies, to use aptamers to serve as my capture biomolecule. So the first thing that attracted me to aptamers, which are synthetic DNA or RNA molecules that can be developed to bind to a wide range of targets, is their flexibility. This was very important, that they have this flexibility to bind to a wide range of targets. In addition, I found out they are more specific than antibodies. And here's the big plus. They are smaller in size. This was really important to me, as it decreases the separation distance between my detection molecule and the sensor surface. Also, they have a longer shelf life, which is another attractive feature. In the work that I will present today, we have purchased our aptamers from OTC Biotech. So the biomarker that I'll be talking about today is C-reactive protein. CRP is known. It's a general inflammatory biomarker and useful in diagnosing inflammatory responses in cancer, cardiovascular diseases, and neurological disorders. In the study, CRP was simply employed as a model biomarker in order to demonstrate the level of sensitivity of our proposed platform. With the flexibility of aptamer technology, we can easily swap the aptamer for our biomarker of choice to be integrated into our platform. Now, given the wide variation in molecular properties, for example, for the detection of protein versus DNA, or in buffer or serum, you require different approaches in order to achieve your desired response. The general goal of the immobilization procedure is the attachment of the capture biomolecule to the sensor surface. Most immobilization techniques involve the first layer of a chemical linker directly bound to the gold, allowing subsequent anchoring of molecules of interest. The overall goal is to engineer a support construct that provides large degree of accessibility to the target analyte while retaining good stability and minimizing capture probe detachment. After choosing your immobilization method, several parameters need to be optimized, such as buffer strength, pH, activation time, depending on the surface chemistry, and your ligand concentration. As shown in front of you, you can directly bind your capture probe. As an example here, they used sialylated DNA, single-stranded, as also we showed in our paper. Or the most favorable method is the use of self-assembled monolayers on the gold surface, which increases the degrees of freedom of the probe and consequently those of binding target molecules. A self-assembled monolayer is created as a foundation of the array comprising of alkane sials, as you can see here, containing terminated amine, hydroxylic, and carboxylic functional groups. After the formation of the sialylated layer, many immobilization chemistries can be performed, and as you can see, here's some examples. A general rule that all types of non-specific binding to the surface, and I stress this a lot, must be kept as low as possible to prevent irrelevant signals interfering with the interpretation of the specific interaction. The use of blocking agents prevents non-specific adsorption. There's a variety of chemical strategies have been reported for the SPRi based sensing, and you'll find a lot of work done in that area. I will now describe to you a general workflow for our experiments. So we start off with a very clean chip, and we perform UV ozone treatment on it, and then use microwave-assisted chemical linker immobilization, and then deposit the aptamers on the surface of our chip using the SPRi arrayer from Horiba Scientific. And then we block our surface with a blocking agent inside the SPRi Lab Plus, which I have currently in my lab, And however, recently, like to point out, recently Horiba replaced the SPRi Lab Plus with an OpenPlex. Now the OpenPlex has the same optical setup as the SPRi Lab. Really, nothing has changed from the optical end, but additional features were added to the OpenPlex, such as the ability to concurrently perform SPR measurements along with fluorescence or electrochemical analysis by simply changing the flow cell. In my lab, we have investigated various immobilization methods because we are working in a complex environment. We chose extra-avidin as a direct chemical linker to the capture probe in order to take advantage of its strong binding affinity to biotinylated probes, as well as its superior anti-fouling properties against human serum proteins. As shown, the first layer involved the formation of cysteamine, which takes about 2-hour incubation, and then activated for 1 hour by glutaraldehyde, where the aldehyde groups bind to the amino groups of the extra-avidin. This last step involves the incubation of the chip in extra-avidin for 1 hour. I forgot to mention, between each step, we rinse our chips and we dry them with nitrogen. And then your chip is now ready for aptamer deposition using the SPRi arrayer. Over the last decade, microwave irradiation has become an alternative to conventional methods in organic synthesis and synthesis of nanomaterials and coating of nanomaterials, offering enhanced speed, reproducibility, and scalability. Now, microwave irradiation utilizes the ability of electric charges present in liquid or a conducting ion in the solid to transform electromagnetic energy into heat, resulting in reaction rate acceleration. I would like to point out that microwave photon is not sufficiently energetic to break or form bonds, but has an effect on rotation as displayed here. We went ahead and compared conventional method of chemical linker modification with microwave-assisted surface functionalization of the cysteamine glutaraldehyde to assess their influence on the binding interaction between the capture and target molecules. In our laboratory, we have a CM Discover LabMate as shown here to your right. The method we established with microwave-assisted chemical linker immobilization reduced the process time from 4 hours to 15 minutes. So the next thing we wanted to do is compare their performances. We deposited on the chip CRP-specific aptamers on one side and a control aptamer on the other side that does not bind to CRP. After injection of CRP under buffered conditions, the change in reflectivity was about 1.3%. However, performing the same test on a microwave-treated chip, we saw a greater amount of CRP binding to the chip. Not only, I'd like to point out, not only did the microwave decrease the procedure time, but also increase the performance of the sensor, and it was very easy to reproduce. So we next examined two different immobilization methods for the aptamer. The one to your left involves the addition of an aminated aptamer to the cysteamine glutaraldehyde surface versus the addition of a biotinylated aptamer to the cysteamine glutaraldehyde extra-avidin coated surface. In this example, we compared blocking agents between bovine serum albumin and PEG, polyethylene glycol. And these are very commonly used blocking agents. And when we looked at their performance, you can see that the PEG provided non-specific binding, controlled more than non-specific binding, but also allowed the aptamer aptamer to bind more efficiently to CRP. Similarly, we saw the same response even when we used biotinylated aptamers as opposed to aminated aptamers. As you can see here, there's also less non-specific binding and more specific binding of the CRP. So we've done a lot of controls, and I don't have the time to show every control experiment we've done, but basically we got to a final construct of our nano-aptasensor, which is composed of where we surface coated with cysteamine, glutaraldehyde, extra-avidin, and then we introduced the biotinylated aptamer specific to CRP. It's chemically functionalized through the binding between extra-avidin and biotin. The surface is blocked with PEG, followed by the injection of CRP. So there are several challenges in directly detecting CRP at ultra-low levels in human serum using the SPRi platform, such as the direct response from the binding of CRP to the sensor surface can be masked by non-specific interaction from serum proteins, which are present in higher concentration than CRP. Changes in the refractive index of the injection solution from the running buffer, and analyte concentration falling below the limit of detection of the instrument. It can be seen that direct CRP detection is incapable of distinguishing between the CRP-specific aptamer, here is black, and the control. To overcome this, we employed a sandwich amplification strategy using nano-enhancers. When I say nano-enhancers, what I mean is aptamer-coated quantum dots. So after we inject the CRP spiked in human serum, we follow with an injection of nano-enhancers to distinguish between the binding of CRP to the CRP-specific aptamer and also not to the control. So here we introduced the nano-enhancer to a pre-injected solution of CRP concentration of 500 nanograms per ml in human serum, which resulted about 18% change in reflectivity, while the control sample had minimal change, around 1.7. In addition, a difference image is recorded to correlate the binding kinetics after injection of nano-enhancers. The spots that have been pre-functionalized with CRP-specific aptamers to your left are intensely illuminated as opposed to the control aptamer spots. The correlation between the binding kinetics and the difference images help further validate the binding interactions between aptamer CRP and the nano-enhancers. Now, to further investigate the reliability of the biosensor in distinguishing different amounts of CRP present in human serum, we assessed the sensor performance with a range of CRP concentrations. And as you can see from our sensogram, a decrease in SPRi signal was observed as the amount of CRP spiked in human serum was lowered. So basically, this is what's happening in the sensor is that after we inject the CRP, we inject the nano-enhancers, and then you can see the actual amount of CRP signal, SPR signal being enhanced here. Now, the corresponding gradient, concentration gradient is plotted where each point represents the average value of the reflectivity difference between initial and final buffer injections calculated from 3 SPRi kinetic curves for each concentration. For comparison, the concentration gradient curve is also plotted for the control aptamer after injections of the nano-enhancers. And you can see there's marginal difference of the signal between the CRP-specific aptamers and the control. The limit of detection was validated to be 5 femtograms per ml. This is an agreement with the limit of detection having a 3-fold higher response than the control. when taken into consideration along with the standard error. Our future work will focus on extending our platform to detect a panel of biomarkers simultaneously with the level of sensitivity described here today. And we're very excited with this level of sensitivity because 5 femtograms per ml translates to 45 times 10 to the negative 18 molar, and that equals to 45 times 10 to the negative 21 moles. So this platform shows that we can comfortably detect these really biomarkers that are very important in cancer, in cardiovascular diseases, and neurological disorders. And the challenge next for us is to extend this platform to look at a range of biomarkers that we can detect simultaneously to better enhance the diagnosis for patients. To this end, I was truly fortunate to work with brilliant scientists, Dr. Stavrizian and Dr. Malic, as I started my journey with SPRi platform, and these are their photos. And now I have the privilege to mentor future brilliant scientists, and I have to highlight that all the CRP work presented here today was done by Stephen Vance. I would like also to thank Brian Quimby for taking this nice photo, and also for financial support. I'd like to extensively thank the earlier.org Friends for an Earlier Breast Cancer Test, and also the Joint School of Nanoscience and Nanoengineering, which is a collaboration between North Carolina A&T and the University of North Carolina, Greensboro. And finally, I would like to thank Horiba for giving me the opportunity to present my work to all of you today. And last but not least, a special thanks to all of our attendees, and look forward to answering your questions. Thank you.
All right, well, thank you, Marinella, for a really nice talk. If nothing else, it's always exciting to hear Zepto used in science. All right, so this is a good one here. Marinella, you ready?
Yep.
Okay. In reference to the DNA detection SPR system, why does your sensorgram show a drop in signal after the injection of the complementary strand of DNA?
Okay, that's a good question. The small drop in the signal following the injection of the DNA target is due to a difference in cell concentration between the DNA stock solution and the hybridization buffer. This results in a negative change in reflectivity for the control. Therefore, the hybridization signal is taken as a difference between the initial and final buffer injection for the calculations of the signal enhancement.
All right, I'm trying to work through. There's quite a few questions coming in, actually.
Okay.
I've seen.
I can't see them.
You can or cannot?
I cannot see them. Yeah.
Oh, okay. So this came up twice here. Let me see if I can summarize it. Why did the aminated aptamer result in a slightly higher SPR response than the biotinylated one?
Okay. Well, there was a slight difference we saw, and when using the aminated aptamer as opposed to the biotinylated aptamer, and we really attribute that to changes in the separation distance between the CRP and the sensor surface. So when you attach the aminated aptamer, your CRP gets closer to the sensor surface, as opposed to having a extra-avided and then biotin and then the aptamer and then the CRP. So that has some influence on the signal.
All right. So there's a couple questions regarding passivation of the surface. For one, well, okay, let's ask this one first. Did you assess your platform with BSA after injection of CRP and spike serum? And what kind of results did you get from that?
Yeah, I didn't show this, but yeah, we of course assessed our platform. As I told you earlier, and when we did the controls, we looked at it in buffer conditions. However, when we looked at BSA with this injection of serum, we had minimal enhancement to signal. And the reason why we think this happened, and they could have been, is that the BSA had more non-specific binding, and thus minimizing the accessibility of the nano enhancer to the target. So it's you know, as I mentioned earlier, it's really important for us when you're designing the system to allow your incoming molecule to have as much accessibility to the molecule on the surface. So we believe it could be that we're fighting two things: is that one, sizes difference in comparison to PEG, the BSA, so that could have been causing some blocking, and also that there's more non-specific binding occurring.
And along those lines, I had a question about the PEG. I know the PEG comes in different sizes.
Yeah.
So how many units were in your PEG?
I can tell you the molecular weight was roughly around 800 grams per mole.
Okay, I think that's generally how they are
Yeah.
how they differentiate between them.
Yeah.
Let's see here. All right, you mentioned ELISA in your talk, and the question here is, how is the sensitivity of SPRi in comparison to ELISA? I think a lot of people probably want to know the answer to this.
Yeah, that's a good question. I mean, on average, ELISA, you can get a good limit of detection at femtomolar. But as you can see here today, we showed with SPRi, you can go down to attomolar very comfortably. And so the advantage is that for me, SPRi is that I can do multiplexing, so I can look at 10 to hundreds of biomarkers simultaneously with that sensitivity. Where I see with ELISA, I would have to buy different kits for each biomarker, so that's where I see the difference.
Okay, and maybe we'll have to give more attention to ELISA in the future. I think that may be a good topic for a future webinar. Let's see if we can get one more question in here before I know you have some other things to do today. This is actually from one of our users. How long do you wait between the injection of CRP and the nano enhancers for the sandwich method?
Yeah, so we generally wait about as soon as about 5 to 10 minutes on average. Yeah.
5 to 10 minutes. Okay.
Yeah.
All right. With that, actually, I have quite a few more questions in here to get through, which means I'm going to have to do my homework and follow up with everyone here. Marinelle has spoken with me earlier, saying that she would be open to communicating with people, so I may have to get you in touch with her. Sorry. With that, let me thank everyone for joining us today. I'll be in touch with you soon. We'll send out some information on this talk. I hope to get a recording out on the web so that you can follow it again at your leisure. Thank you everyone for joining us today. I hope this has been a successful talk and that you've learned something from it. And you should hear from us soon about future webinars.